over the last decade, we see an increasing rate of GWAS studies. This is because people really want to see, based on my genetic information, can I predict a disease? How likely am I going to have a disease? How also they are interested in whether there's association with the patient's response to drugs. And this is facilitated by the drastic reduction of genome sequencing cost in early days by the microarray cost. And so you can see the number of publications in the GWAS studies are really increasing. In the early days, when the cost is high and people didn't have enough data yet, they also it's kind of collecting sample cohorts is very time consuming. You take decades to collect enough patients for some case control studies. And so in the early days, they were looking at cohorts that have only a few hundred patients. And so sometimes they might find a SNP that give you some association. And if they repeat the same study, again, using another scientist do the same study, they collect another cohort of patients with a few hundred patients, they do the GWAS, they see some SNPs, and there could be very low overlap between the two studies. And this is mostly because in the early days, there were not enough patients. Most of these GWAS individual association have very weak effect to the disease. And because we're doing so many multiple hypothesis testing, you actually hit on things that are not really real. And if you change a cohort, you'll land in some other regions. And so you can see here, the number of GWAS hits is directly associated with your discovery cohort size. Early days, it could be a few hundred or a few thousand, and then to 20,000 and even 200,000. And now with more and more people doing genome sequencing, you might even have these huge cohorts that have close to 1 million individuals to look at association. The nice thing about this is, once you sequence somebody's DNA, the information is always there, and all you need to decide is which phenotype do I want to look at. You can see here, this could be height, body mass index. This QT interval is heart QT interval. Cholesterol level, you can look at hair color, eye color. You can look at disease, other association. You can look at their IQ. Yeah, it's just many like skin color and things like that you can look at. So in these type of GWAS studies, to make sure that you are getting the robust result, having bigger cohort is very, very important. Another important thing is to make sure that you don't have population stratification. So for example, if I want to look for SNPs that can predict longevity, I collect the people who can live over 100, centenarians who are over 100 years old versus people who died before they reach 100 years old. And, of course, centenarians are hard to find, but you collect those and you do the GWAS study, and you might find, it turns out, hidden things you don't know is the centenarians are enriched in, say, this Japanese population. Whereas the people who died otherwise younger are kind of a random mix. And so because of that, when you are looking at the SNP difference, what you might see is that, oh, the Asian SNPs are all showing up because it has nothing to do with longevity. It just happened that in your cohort, the longevity cohort have more people from Asian or from Japanese. And so in this case, in your case, you have an overwhelming population, whereas your control is different. And so what happens is that when you do the p-value calculation of different SNPs, remember we are doing this for millions of chi-square tests, we can look at the p-value of those chi-square tests and then do a QQ plot to compare the expected p-value and the observed p-value. If you don't have population stratification, you would say that, okay, most of these SNPs are not significant. Then suddenly we have some SNPs that are significant these are real. Whereas in other cases, if you see kind of a very early diversion from your expected, like this is an indication that your case control populations have this population stratification. There are a lot of SNPs that are significantly different between your case and control. That could be because of racial, ethnic groups that are differently populated in your case and control. And so for that, you have to correct that and kind of only call those ones as your real difference. Another potential, so this is one example. If you also just run the PCA on all the SNPs, you can already see that on this PCA plot, there are people from different populations. And so, correct for the population difference from your case control studies that are arising from the uneven populations in the different cohorts. Another situation is there are some unknown relatives. Maybe you have a distant cousin in this study that you don't know, or in a case control, we don't want family structures. And so you really want unrelated family members for the case control study. And so there are also something called IBD, which kind of give you the chance that these two individuals are from the same family. And so if on the IBD test, you see, you see a lot of people from the same family, you might want to remove the sibling or the cousins from the study. You really want a representative member from unrelated family in your study. And so by this time, for example, this is a figure in 2019. In NCBI, there is a GWAS catalog. And they continue to collect published GWAS studies and get their SNPs from them to see, this is on chromosome 1, 2, 3, 4. What are the current discovery of SNPs that are associated with different diseases? And so there are like many of those summary statistics files that tell you this GWAS study identified the following SNPs as associated with that disease. And another is a different collection of SNPs association with disease. And based on our lecture in the last Hi-C interaction lecture, Dr. Feng Yue mentioned that a lot of these GWAS SNPs, people found that only a very small set of those are actually in coding regions. Because coding region is only 2% of the genome, a lot of these GWAS-associated SNPs lands in non-coding regions of the genome. And interestingly, a lot of them are enriched in DNA's hypersensitivity peaks. Well, or there could be some that are not in the peaks, but in perfect LD with those peaks. This is because sometimes they didn't do genome-wide association using genome sequencing. They just use SNP array. They typed one SNP, but they don't have the SNP in the DNA's peak region. But these two are perfectly linked, so you are suspecting that the real effect is on the DNA's hypersensitivity region. And only a quarter of them are not in LD, which suggests that a lot of these SNPs associated with disease are related to a transcription factor that binds to a region which influence the nearby gene expression. This nearby could mean 50 kb or 200 kb or something like quite a distal region that influence the expression of that nearby gene, which then have an effect on the phenotype. And so understanding epigenetics now become very, very important to interpreting these GWAS SNPs. It's like, oh, there's a SNP in here, it's associated with a disease, what's happening? We want to see what transcription factor binds that enhancers and what kind of nearby genes are linked, and then we can understand why that's related to the phenotype or the disease. Another type of study that's really useful is called eQTL analysis, and a national project, a consortium project called GTEx is really, really quite exciting. So first, they look at 1,000 individuals. These are not disease or no specific disease. It's just average people who have died from a lot of them could be unnatural causes or whatever. And so they donated their body to the project, and they take different tissues. In this case, brain, heart, like many different tissues, 50 different tissues from these individuals. And they did both the SNP typing of, this is using SNP arrays to look at their SNPs across the genome, and also to do the expression of those 54 tissues across individuals. And so in here, because we are not interested in disease per se, the trait in here is no longer whether this person have blue eyes or black hair, or whether they have a fair skin, or whether they have high body mass index. Instead, we use expression data as the phenotype. You say, well, it's 1,000 people's liver, but for this gene, it's expressed at a much, much higher level in some population, but lower in another population. What is the difference? And so the features are on the SNPs. And we say, oh, it turns out if they have, in this case, for example, we say if they have these SNPs, the expression is much, much higher in, say, liver. But if they have another SNP, the expression of the nearby gene is happening much lower in liver. And so you can establish this association from something called expression quantitative trait loci. So quantitative trait is, height is more quantitative. It's not a black-white 1-2. It could be just the expression level. And the trait in this case is not really eye color, physical trait. It could just be an expression. And we are trying to look for a particular SNP locus that's associated. The SNP in this locus is associated with a quantitative gene expression level. And using this type of eQTL analysis, again, a lot of people find that there are SNPs in nearby regions that are linked to gene expression differences. And a lot of those are also in the ataxic or DNase peaks, which means that gene transcription factor binding in the distal regions might influence this gene expression and eventually influence real phenotype or trait. Yeah, so that's kind of the summary. In the genome, there are SNPs. They are often linked if they are in proximity on the chromosome. And there are also these haplotype blocks where a lot of SNPs are linked to each other. And in order to really do the genome-wide association studies, you can collect big families with members associated, are known to be associated with the disease, to look at the allele that's transmitted, which made the difference. Or you can just do case control studies, normal versus disease, and check every allele to see whether there is any difference between the allele frequency in the two populations. And in order to increase the statistical power of your analysis, you can look at haplotype association rather than individual SNP association. You could increase your patient sample population, bigger cohort, the better power. And also, you need to make sure to remove the populations, eliminate the population stratification, and remove the too close family members. This is in the case control studies. And also, we can use the eQTL to identify SNPs associated with differential expression. And also the GWAS SNPs are also linked to some trait. And a lot of these eQTL sites and the GWAS SNPs are in the non-coding regions. And then we need to use the epigenetic data to help us understand what's happening. Okay, so that's all for today.